So in the first part, we talked about fungi and how, as a more, general perspective, how to encourage them to come. We explained many reasons why that is good for us, for the ecosystem, for all life on Earth. Now we'll go a bit, we'll become more scientific. So we'll split and look at specific functions, but always keep in mind that the categorizations we're doing are just human ways of trying to understand what's happening. Because I'll mention that many times during this session. Many of the things that we are saying, like this fungus under these conditions is doing that thing, are true most of the time. In other times, in other conditions, they can do something completely different. So they're multifunctional, but what we usually see is their habitual ways of being. But, habits are 90% of the time, but some other times you do something else. So keep that in mind, that they're not specific, they're not absolute, let's say, they're not dogmas, what we're talking about here. And I'll be mentioning that. So we'll see the fungi in the soil that we have now encouraged and we have created the conditions for them to thrive. What are they doing in there? We'll concentrate mostly on mycorrhiza, fungi that grow on roots, but we'll talk about a few others as well. So, many different ways to categorize fungi or any other lives. Like sometimes we categorize them morphologically, in other words, how they look. Other times we categorize them functionally, what they do. And two things can look alike but do something very different. So now we start with a functional categorization. So there are a few ways. I just got hot. So there are different functions that perform. These are a few of them. And we'll just go through these, but we won't get too deep in most of them. As I said, we'll go deeper only in a couple of them. But just to have an idea. Plant pathogenic, they are fungi that we see as disease, and we have categorized them as pathogens. Lichenized fungi are the fungi that we said before that live together with cyanobacteria or algae, and they create a little tiny ecosystem. They are one of the pioneers. They live on many things. Zygomicota are molds, the things that you see on moldy food. Yeasts are the things that help us mostly to create a lot of kinds of tasty food, like soy sauce or bread. Endophytic are the ones we mentioned in the introduction. They are actually fungi that live inside phyta, plants, and they perform a lot of functions and they help the plants. Aquatic fungi, this is one of the latest developments. The last few years we have started discovering more and more aquatic fungi. Basically, fungi that exist inside water, even in the sea there are fungi. How they do it, we have no idea. We're just starting to understand, we're just starting to discover them, let alone understanding how they function. Saprobiotic, they have another lifestyle. They actually, sapro means rotten, biotic, their life. So they live by rotting stuff and they break down dead organic matter, bringing it back to life inside them and inside a lot of other organisms. And mycorrhiza are the ones we said that are living on the roots of plants, and they have a really close and very mutually beneficial relationship. Pathogenic fungi, they can actually, as we said, they can grow on the leaves, on the stems, inside the trunk, or on the roots. They can grow anywhere on the plant, depending on the species, and they the result is usually they make them sick or kill the plants. And very often we try, as humans, because we're very close-minded sometimes, we try to eliminate them and kill them. So we use, for example, fungicides, if we see a fungus there, and we go and spray, and probably dies this fungus, but along the line, also many other fungi die. And then you have a population of plants that dies, and you say, but I eliminated this pathogen, why did it die? Because it didn't have the other fungi to support it. Yeah?
So you mentioned the fungicide. Just curious, what's the main mechanism of fungicide?
There are many.
Okay.
There are many, many different ways. People have come up with amazing ways to kill fungi. Many different ones, from starving them to dissolving them, a lot of different stuff. Not very useful, unless something grows inside you or on you. But also, apart from the fact that, I'll say that again later on, and you'll see specific examples of that, we have realized that a lot of the things that we call pathogens, as we were saying in the introduction, are actually organisms that perform a function, but if the ecosystem they live in is disturbed really heavily, they go out of balance and they occupy the vacuum that is created by killing something else. And they become pathogenic. It's not like their life purpose to kill other stuff. They become pathogenic because they are too many, like humans. Now we're becoming pathogenic because of our behavior. So they can kill entire populations. Sometimes they have a function of killing entire populations in order to mediate the plant succession. So a forest has reached a point where it has become almost a monoculture, for example, it's beech forest. A fungus can come and can kill big part of it so other species can thrive later on. And that's a function. And sometimes humans don't understand that. They would try to keep back succession, for example, by trying to manage nature, as if we knew better. Saprobiotic fungi, we've talked about them before many times. Just a quick reminder, they grow beside or on their food, and they excrete the digestive enzymes. They break down more complex, long chains, molecules. Who's hiding? You. They break down complex plant and animal residues into simpler molecules that they can then absorb by the hyphae and, keep on growing. Mycorrhizae now. Mycorrhizae are these amazing organisms that they were first discovered by a forest pathologist in 1885, long time ago, but it's only the last 30 years we have started to understand them and better comprehend what exactly is going on. I would say actually even the last 5 years, we have discovered some of the mechanisms, not understood them, but we have discovered amazing mechanisms we'll be talking about today. It's very, very recent when, even if we know them, we don't understand how they function. So they basically colonize root tissue in different ways. They have different lifestyles, and plant-produced carbon and lipids actually. Plants produce lipids like fat substances, and they exchange them for fungal acquired nutrients. So, to see here, a plant without any colonized roots, it has a much, much smaller surface area to accumulate nutrients and water from. The mycorrhizae, they come in and they get connected to the roots, and it's as if they have the extensions we're talking about. So they have a much larger area to absorb all these nutrients. Not only the area, but they have different mechanisms. The mycorrhizae can absorb nutrients that otherwise would be completely unavailable to the plants. So 89% of the plants, they form relationships with mycorrhizae. They must know better. I mean, they do it for a reason. Because they thrive, they grow better. Some, as we said before, would not even be able to survive It's not because it's better, it's because it's essential for their survival. There is an 11% that probably, as we were discussing before, they developed in areas where there were different conditions, or environmental conditions probably. We don't know why, but it's a hypothesis that they developed and evolved there where there were limited resources or different kind of resources or too many resources. That's one thing to keep in mind. If there is one thing to keep in mind, I would suggest this is the one. If, about mycorrhizae, if things grow in very rich soil, whether rich, very nutrient-dense soil, whether that's because of inorganic salts that use as fertilizer, or whether that's a heavily composted land, then most plants, they do not associate with mycorrhizae, even if they can, because they don't need to. Say, why should I waste 30% of all the sugars I'm producing to give it to mycorrhizae if I have loads and loads of nitrogen and loads of phosphorus, for example? So keep that in mind, that more is not always better, because the mycorrhizae don't just give more nutrients to the plant. They give a lot of other stuff, as we will see later. And I'll repeat that again, because it's very important. More is not better. Yes?
But if they don't associate with mycorrhiza, can they absorb the nutrients?
If you give them compost with a lot, or salts, like phosphorus salts, then if they dissolve in water and they can absorb them, yes. Okay. Otherwise, they'll have to mine them, and it's mycorrhiza that mine them. Okay. But the thing is, in most farmers, at least minds, it's either plants alone not being able to absorb nutrients in a poor land, or plants with fertilizer and water-soluble salts that they can absorb them. The in-between is the answer, actually. Yes?
You are talking about plants that have evolved for nutrient-rich soils, or that have been nurtured in nutrient-rich soils?
Both. Both. They don't need them. And I'm not talking about the 11% that doesn't associate with them. I'm talking about plants that would otherwise really love to go for them. So again, keep that in mind. I say it again and again. More compost, for example. More fertilizer is pretty obvious to everyone, inorganic, but more compost is not better. There is a balance there. Because I've seen it again and again. People who have very heavily composted gardens, and then other things come. For example, if you have too much nitrogen, even if it's from the compost, then the plants will grow leaves really fast. Their cells won't be strong. The cells on the walls of the tissues. And then many things can enter very easily in there. They're not strong. They grew too fast. One example. Another thing which is important to have in mind and look into it, you know, nitrates. Nitrates are used as preservatives in salted meats and stuff like that. They are cancerogenic. Actually, we have done, if you're an organic farmer, you're not supposed to do these measurements, but there is a lot of organic food that has huge amounts of nitrates in them because of all the manure that is being put, non-composted manure. Okay, so again, there are things that we can do to help the soil, help the plants, and at the end, we're not helping the plants or ourselves. Keep that in mind. Okay. Yes.
Sorry.
Tell me.
It just reminds me, like it's like treating symptoms instead of treating, like in different types of medicines, like.
Exactly.
Yeah.
Exactly. Instead of building, like at the end of the day, no matter what question we have or problem we have, the answer is biodiversity.
Yeah. And to build.
Always. If the answer is more phosphorus, it's not correct. More of that, more of the biodiversity is the answer. Yeah. But there are steps and methods to encourage that. And you might not see tomorrow the effect, but you'll see it after a while. So, let's concentrate a little bit now on mycorrhizae. Now, these categorizations we're doing now is not functional, but it is morphological and anatomical. So when we look at them, they look different. Okay, or also some categorizations are plant dependent. In some species associate only with this type of mycorrhiza guys. I don't like the other ones. So they might be sharing functions, all of these kind of mycorrhizae, but they look different. And that's important though. So just to keep them in mind and understand what they mean, we have ectomycorrhizae. Ecto is from outside in Greek. Exoskeleton, outside. Ectendo, it means it's outside, and endo means inside. Outside and inside, they do both. And endomycorrhizae, which is inside. What does it mean, outside, inside? It means that they are associating with the roots. The endomycorrhizae go inside the root cells. They penetrate them. The ectomycorrhizae, they just cover them. They grow around them. The ectendo, they do both. Endomycorrhizae, there are different types depending on what kind of plants, what genuses of plants they associate with. Ectomycorrhizae are actually descendants of the saprobiotic fungi. So imagine saprobiotic fungi, which are actually breaking down organic matter, and they absorb it and they eat it. But now they have evolved a little bit more and they are able to go into the plant and give it some of these nutrients while they're getting sugars. So they're still saprobiotic, but they're also able to associate with them. Interestingly, we have found out that many of these that we thought they could only survive and exist with the associating plant, they can actually start growing. They have hyphae, they are saprobiotic, they live like that until they find their plant. And when they find their plant, they associate and then they fruit. So you will not see them if you're looking on top of the soil. They might exist there until they reach a plant, or you go and plant a plant. Until then, they're just living as saprobiotic. So these ones mainly decompose nitrogen-rich polymers, so they're able to get nitrogen as well and can give it to the plants, which is great. They absorb water and other micronutrients, and they get the carbon. This is what they look like. They basically form a sheath of hyphae around the roots. This is an actual root. You'll see a few more photos. They're really, really impressive. They weave their hyphae around the whole root, and they enter inside the epidermis, but they do not enter the cells. They're in between the cells. They're covering them. This is an actual electronic microscopy photo, and you see this is a plant cell. These are the hyphae. They just go all over the place around them. And this is where they get, through the exudates that our plants are giving, they get the carbon, and they depose all the other things, water and minerals, between the cells. So actually, when these go well and it's in a good fungal soil, they can be up to 40% of the root mass, which is crazy. It's like huge, huge mass on the roots of hyphae exchanging nutrients. So here it's an interesting, I don't know if you want to get into that, but for me it's very exciting. The mycelium penetrates between the cells of the root and they exchange. How do they exchange the nutrients? There are these kind of proteins. I'll just say it very, very quickly because I really love it. So in the cell membrane, there are little canals that are usually closed. And there are some proteins that both the mycelium and the plant have constructed that they're transport proteins. So imagine, for example, a protein that is like this. Let's say it has this shape. The canal is here. So here is the cell membrane. Here is the mycelium around it. And there is a protein that's like that. This protein cannot fit over here, right? It cannot go through this canal. So when an ion of, if you don't know what an ion is, doesn't matter, a little atom, let's say, of a nutrient is released by the mycelium, let's say you have calcium over here, then it goes and attaches itself over here. It has a receptor, this protein has a receptor, like a hook that is attached to the calcium ion. And another one attaches itself over here. And so these two sides that are positive and positive, what happens to them? They repel each other. And from this, now, this protein has become like this. Because it's positive here and it's positive there. And now it can actually fit through this canal. It's a very evolved kind of mechanism that they have discovered in order to communicate the mycelium with, they have developed the mycelium with the plant cell. And this way they make canals specialized with the proteins so that only these things can go through and not random pathogens, for example. This thing can fit only when this actual nutrient attaches on this protein.
The protein is produced by the hyphae or by the...
Both are producing these proteins because they're exchanging stuff. There are proteins, transport proteins that for all kinds of nutrients.
So this protein, for example, is specifically for calcium ions?
Exactly. There are some proteins that are more, but for calcium, because I have read this specific one, that's why I'm giving you this example. But there are proteins that are more general. There are proteins for more complex molecules as well. But this is the general, like simplified version of what transport proteins are.
That's for the game, that there's like a semisphere and you put stars and circles and squares.
But the funny and interesting thing is that these stars and circles, they change shape depending on the needs of the plant and what the mycelium is bringing. And they change the shape and suddenly they can fit. So this one is an example, and a real photo, of Picea glauca. This is a fir that grows in Northern America with a Lactarius. Many probably have eaten Lactarius, which is the fruiting body of this amazing structure on top of a piece of fruit of this fir. It really holds it, like embraces it and protects it in many, many different ways. That's another example of an ectomycorrhiza, Amanita muscaria. We've all know this lady. And on a Pinus radiata, another Pinus conifer. And you can see how it has completely covered some of the roots. Another photo, just to see how it's a Chuga canadensis. This hyphae that goes around, you don't really care about it, but I like to share this information. It has a specific name. These hyphae that are embracing each and every cell, they're called Hartig net hyphae. This net has a name. Hartig was one mycologist, and in his honor, they named this kind of net. So, ectendo, very quickly, it's a new category. The last couple of decades, we have realized that, there are some that do both. They actually embrace, they create this Hartig net, but they also sometimes get inside. So we came up with this ecto and endo. They do both kind of things. These are some of them. I really want to, this one my favorite that I was, it's really amazing. Orchids, we said about orchids. They have, as we said, there are 22,000 species. They have a huge compatibility with thousands of different fungi. And initially we thought, well, what happened? So the seed of the orchid germinates. And instead of actually germinating, very often you've seen microgreens, right? Or you see the little leaf coming up, and after a while there is a root coming up, or a little root and then the leaf. Well, the orchids, because they rely on mycorrhiza to survive, there is no leaf coming up. Just roots from everywhere, from all directions. Tiny little roots growing from all directions. They're trying to find mycorrhiza there, because that's the only way they can survive. They try to find mycorrhiza, and many actually do the favor to the orchid roots, and they start associating with it. So basically, this mycorrhiza, they enter inside the root cells, and there they create a little bubble. And this little bubble that is filling up with nutrients and it's filling up, at one point it bursts. And then all these nutrients is another mechanism, very different from that one. It bursts inside the cell, and all these nutrients are now absorbed by the actual cell. And then, because there are so many different types, they take turns. So, because one mycorrhiza can be more efficient at bringing in calcium, another one nitrogen, etc. So they take turns, and they're feeding this little baby orchid one after the other. There's a community that is actually feeding a plant to be, because it's not yet a plant. This state can actually last for orchids a few years. It's really crazy. For years, it can live underground, just fed and nurtured by mycorrhiza. And when it's ready and has developed a pretty good root system and a lot of relationships with mycorrhiza, then it will start sprouting and going upwards to have an actual over-the-ground system with leaves and flowers, etc. It's a very special kind of plant, very, very, very evolved, because it creates mechanisms that, it shows a lot of resilience on one side, but on the other side, many orchids are endangered, as you might know, because of the deteriorating situation in the land. Because as we saw, as you go backwards in the succession with human activities, then the first ones that start disappearing are fungi. So orchids are endangered because of this. Let's go to the next one. We have these other four kinds of endomycorrhiza. Cavendishioid mycorrhiza, they're a pretty recent addition. I think 15 years ago, they discovered this type of mycorrhiza in tropical forests. So you see here, they have different styles and architectures of developing. So these ones, they do the network, the hyphae surrounding the cells, but they also get in and they make a helix like that. They're also, you could talk for a long time about all them. Arbutoid, they associate with the Arbutus genus, strawberry tree, Koumaria in Greek. They associate with these guys in pretty high pH environments. They can survive. Monotropoid, another amazing family of plants. They are actually plants that have no chlorophyll. They do not photosynthesize. So how do they live? We used to think that they are parasites, but actually they're not parasites. They are very efficient at absorbing other kinds of nutrients. But they don't have sugars, such as the mycorrhiza, right? They do the same thing. They don't have chlorophyll. But through the monotropoid endomycorrhizae that grow like this, they are able to associate to other plants. So the fungi come in, they associate with these plants, the monotropoid plants, and then on the other side, they go associate with plants that do have chlorophyll and they produce sugars. And they transfer the sugars to the monotropoid parasitical plants. But at the same time, they're not parasitical because they're absorbing other nutrients that they're giving back to the other plant. So they're basically, these kind of fungi are middlemen between plants. Another type is the ericoid. Ericoid is a big family of plants that we have here as well in the mountains, but they are able to survive, some of them, in extreme conditions, like in very northern, close to Siberia, Arctic Circle plants, where the pH can be 4.5, extreme. It can be almost bare rock, extreme temperatures of minus many, 50, 60, and they're able to survive just because of the mycorrhiza. These mycorrhiza are making it possible for plants to exist in areas where it wouldn't be able to support any other kind of life. So that's how the now we said the endomycorrhiza, they just get in, through, around the cells and inside the cell, and there they make a little tree-like structure. And that's why we call them arbuscular. Arbuscular, arbre in French, arbus in Latin, árbol in Spanish, it means tree, right? Because they look like trees. It's a phenomenological, it's a morphological kind of description of this mycorrhiza. And you can see here how they are really taking over the cell, but without harming it, by feeding it. So, what are the main differences between ecto and endomycorrhiza, which are the main categories? The ecto can reproduce sexually, which means two different spores, they sprout close to each other and they start growing, and then they come and fuse this hyphae, they fuse and they grow together. Sexual reproduction, DNA from different individuals. And they start looking for a host plant. But they can live, they can survive saprophytically. They keep on breaking down organic matter, they eat it, they grow, but looking for a plant in order to get some sugars. On the other hand, the arbuscular mycorrhiza fungi, the endomycorrhiza, they reproduce asexually. They produce no above the ground fruiting structures. These ones, the ectomycorrhiza, are most of the mushrooms, not some of the mushrooms that you see, like boletus, for example, the chanterelle, and many of the edible choice mushrooms are ectomycorrhizal. They create fruiting bodies. The endo, they don't create fruiting bodies, they just create spores. At the right time, when they're ready to reproduce, and we'll talk about more in the workshop in the afternoon, they produce a spore, and that spore, again, when the conditions are right, it sprouts and looks for a host plant without needing a partner from another plant in order to fuse together and have a sexual reproduction. Very different kind of systems. Endomycorrhiza, they produce hyphae, we said, inside the roots, and ectomycorrhiza outside, around the cells. The arbuscular mycorrhiza, the endo, and the ectomycorrhiza, they employ different nutrient acquisition strategies. The AM fungi, the arbuscular, they scavenge for nutrients that are released by saprophytic microbes, ready-made there. They don't break down stuff, they just absorb stuff really well, while the ectomycorrhiza actually break down more complicated chains, and they mineralize them. Mineralizing means they absorb them in their bodies and they immobilize them. They keep them inside from organic matter, and this way they access some forms of organic nitrogen directly. So, again, more geeky stuff, maybe you don't care, but it's really nice to understand how the arbuscular mycorrhiza and the plants, they hook up. How do they know that this thing that is coming inside my cells is not an enemy? Actually, the AM, they produce this long word. It's lipoketone oligosaccharides, fat, ketone, ketini, like the coat, oligo, little, and saccharides, sugars, which is a signal that the plant understands telling them, look, I'm coming into your cell, but I produce this stuff and I'm a friend. I came here to help. The parasitic ones do not produce that, so the actual plant is starting to fight against them. But this is one of the signals that telling the plant, I come here with a good purpose. So this way, it's triggered to start a symbiotic relationship, not only allowing the colonization of itself, but also starting to give it food. So why do they hook up? We saw how, but why? The mycorrhizal hyphal exudates, organic acids, and many other things, they solubilize minerals that would not be accessible to plants. Also, as we said already many times, they have a very efficient way to suck up water. That's why fungi need a lot of water, but they are able to suck up water from a huge volume of soil and in a very efficient way. And they actually can share it with their host plant. Also, and again, these are very few reasons of the many, many things we're starting to discover. Also, they are producing auxins. You know what auxins are? They're hormones that plants regularly produce in order to trigger growth. And the mycorrhiza comes there and it produces these auxins, the gibberellins and cytokines and many, many other things in order for the plant to grow. Why are they doing that? They're not, of course, they're nice guys and they're helping, but also the more the roots grow of the plant, the more space they have to grow as well. So it's a benefit for them. Take this hormone, grow your roots, take these minerals, grow your roots, and then I have more space to grow as well. More of us can exist over there. As we already have said, but to stress it once more, they make a lot, they help the plants to be a lot more drought tolerant. The extension of the plant root system is increasing tenfold, 20-fold with the mycorrhiza on them. So they can increase the water absorption. We already talked about cacti and all desert plants that they wouldn't be able to survive in most places. Also, they can stimulate the expressions of genes involved in drought tolerance. So, I think we talked about with Luca, where is Luca? There, about the expressions of genes. So, many of us and plants and all living organisms, we have genetic information inside us that is not expressed, it's not triggered. Some genes are not active. So, arbuscular mycorrhiza have learned how to help the plants by promoting through molecules that they produce the expression of specific genes that are helping these plants to cope with drought. For example, we know that there is a specific hormone that the plants are producing in order to close under extreme situations that happen suddenly. Let's say the temperature is increasing from 20 to 40 degrees. So they close their stomata. We all know the stomata? The little mouths that the leaves have in order to breathe from there. So there is a hormone that is produced by the plants and these stomata are closing so they don't lose water. It's like our pores are closing down so we don't lose sweat if we are really thirsty. Recently there was a study. Until now we thought that the mycorrhiza are actually producing this specific hormone and the stomata were closing. Very recently there was a study with olive trees actually that they had been inoculated with mycorrhiza, that they saw them under extreme stress, artificially made, that the stomata were closing only in the inoculated ones, but not in the non-inoculated ones. And they thought, okay, it must be producing this hormone because that's the mechanism we know. And they found out that this hormone did not exist. So the mycorrhiza have other mechanisms to regulate the functions and metabolism of the plants that we have no idea about. We're just starting to discover all this stuff. Another thing, yes?
we're talking about mycorrhiza and plants, but are there also fungi to fungi mycorrhiza type of connections when we're growing mushrooms?
Well, mycorrhiza is the symbiosis of the
Specifically.
But the same type of symbiosis.
So
Obviously not.
symbiotic fungal relationships. There are many. There are many symbiotic fungal relationships between also different families of fungi. So, like yeast can help actually saprobiotic fungi, and the other way around. So yeah, there are symbiotic relationships between them. Of course, they can be antagonistic as well in specific environments. So another thing that we have discovered very recently that the arbuscular mycorrhiza are doing is that they are increasing the polyphenol production. Polyphenols are some of the most important substances that exist inside plants that give a lot of their properties. Like in olive oil, for example, when you're measuring the quality of olive oil, one of the most important things is the polyphenol content. And we have found out that inoculated plants, they skyrocket the amounts of polyphenols in the tissue of the plant, whether the fruit or the leaves, etc. And of course, they do that not for the humans who consume it, but for the plant itself, because it has a lot of benefits and it builds up its defense. But humans who consume it, they have a lot of other benefits, such as antioxidant activity, anti-inflammatory effects, cancer prevention, neuroprotective effects, and botulation of the gut microbiota. So all this stuff we get as benefits just because we went and we inoculated our plants with mycorrhiza. And apart from the fact that our plants are happier, we as humans are much, much healthier. Other kinds now, let's leave the mycorrhiza aside for a second and talk about other applications really quickly. Biocontrol of soil pests. Some are very obvious why this happens. Actually, there are fungi that can attack nematodes, as we saw in the introduction, and they can really control the populations, and other root-feeding insects, either by trapping them or by infecting them and killing them, like the cordyceps that we have seen yesterday. There are specific fungi that are entomopathogenic. Entomo is insect in Greek. Pathogenic is that it creates a disease. So there are fungi that are disease, they're pathogens for insects. So we can use them and encourage this kind of fungi to grow in our land, in our soil, so that specific insects that can get out of balance and have too large of a population, they can be killed by the fungi. Another way is mycoremediation. We talked a little bit. I have quite a bit of material here. I won't get too much into it, but there are so many applications of bioremediation with so many different ways. Petroleum hydrocarbons, mycofiltration through the hyphae network. You can keep a lot of heavy metals that they can suck them up, and even pathogens, they can filter pathogens. Decomposition of agrochemical residues, we have already seen that. Other types of mycoremediation with Lacasse producing fungi. Lacasse is actually different types of enzymes that oxidize a lot of chemicals. They have actually right now being used in the synthetic dyes industry. They have the fungi that produce this enzyme, and it breaks down these very harmful synthetic substances. Decomposition even of chlorinated compounds. They break down chlorophenols and chlorinated solvents, very harmful as well. You can remediate soil that has been polluted by that, or groundwater. In the pulp and paper industry, also you can use them in order to break down a lot of the pollutants that end up in nature. You can use that before they actually are disposed in water. Another application that I have read a paper about, which is very exciting, I find, is using, we talked about how with mushrooms you can suck a lot of heavy metals, but you can also use, many plants cannot survive in polluted, heavily polluted with heavy metals soil. But we have found out that with mycorrhiza, they can survive and they can actually absorb a lot of these heavy metals. So the mycorrhiza absorb them, they give it to the plant, and instead of the plant dying, it can actually still develop because it gets a lot of other benefits as well. So there was this experiment with Tithonia diversifolia. It's an amazing, my favorite plant for organic matter. It's insane. You can cut it down to the ground 3-4 times a year and it will grow 3 meters high. It's crazy. So it's perfect for this because it grows a lot, so it sucks a lot of nutrients, so it sucks a lot of heavy metals. That's why they used it. So they put them in very contaminated soil with lead and zinc, and the ones that were inoculated, they got reduced by 20% their growth, but the ones that were not inoculated were reduced by 50%. So it was more than double the overall growth, and also the ones that were inoculated, they had 50% more lead and zinc in their leaves. So what we were saying the other time, you cut them and you bring them somewhere else and you can extract these heavy metals to clean up the soil. Also, we said about that as well, they create aggregates. In other words, they excrete this glue and they glue all the soil particles together. They help controlling erosion. Many lichen species colonize the soil surfaces actually in very bad degraded land, rocky substrates, and this way they help stabilize the soil and prevent erosion. A lot of endophytic fungi, we have talked about them also. They live inside the tissues. They help the plants resist different environmental stresses. They improve. A side effect of that is that some grasses, for example, can survive in very difficult situations and thrive better, so they have a bigger root system, so they stabilize the soil again, especially in grassland ecosystems. They act as a binding agent, all these hyphae inside the soil. I'm just going through them quickly because there is a lot. And as biofertilizers, because they can the nutrient uptake can be much higher. There are specific examples here. I don't need to go through all of that. Also, as biocontrol agents, one of the ways they do it is actually, you saw these photos of these roots that they were completely engulfed in hyphae. Well, if you're engulfed in beneficial hyphae, other pathogens or insects cannot get in. You're completely protected. It's a shield actually around the roots. That's one of the main mechanisms, but there is a lot of others. Many fungi produce antibiotics. Boletus and Clitocybe species that we know and we eat, they produce antifungal substances for other pathogenic fungi. Another new exciting field that we just start discovering is this, what we called induced systemic resistance. So the exact mechanisms are quite unknown. So we make experiments and we see the effects of that, but we don't know how they do it. But what we see is that just by having mycorrhizae, suddenly the plant itself is producing different substances and changes in metabolism in order to resist different kind of pathogens. There are a few examples. You can go through that. Some examples. We tried Fusarium oxysporum is a big problem with tomatoes. It's basically really quickly within days, 2 or 3 days, your tomato plants can wilt and die. So inoculated tomato plants with AM fungi, they showed a lot higher resistance to Fusarium populations. The Fusarium populations did not decrease. It doesn't mean that the mycorrhiza killed this pathogen. They're still there, but the plant resisted much better. Same with beans and the Rhizoctonia solani. It kills the roots. They've made experiments, and it can resist much, much better cucumbers and other kind of pathogens. But these are examples of the induced systemic resistance, where the plants, just because they are in association with mycorrhiza, they are much more resistant. So another thing that I have mentioned, but I would like you to keep in mind is this, because this changes the way we see the ecosystem and the world, I think. I repeat it just to stress it. In many recent experiments, things that we thought that were pathogens, they proved to be protective organisms. Like, go behind, the one before, the Fusarium oxysporum, big problem with many plants. It has been actually shown to reduce the populations of this Medidogene incognita, which is it is a nematode. So, while when it is out of control, it can actually destroy our plants, when it is in a balanced population, it can actually protect our plants from nematodes that are actually eating the roots. So it's becoming pathogen again, I'll say it again and again, it's becoming a pathogen when it's out of balance. When it is in balance, it is actually protecting us. And this is where a big problem with agrochemicals is coming in. When you go there and you have Fusarium and you spray and you kill it, and you kill a lot of other kinds of stuff as well, a lot of other fungi that are beneficial, but even the fact that you kill completely the population of Fusarium, suddenly you have a nematode problem. And you go and spray the nematodes and you kill the nematodes, and suddenly you have an arthropode problem, because the arthropods are killing the nematodes and now they don't have any food, so they either go and eat something else or they disappear, and the chain of problems is becoming bigger and bigger and bigger. So you're patching the problems one after the other, and you create a myriad of other problems. And this again, that's why I said the answer to every problem is biodiversity. No matter what the issue is in your garden, in your forest, in your ecosystem, even in human societies, I believe, it's biodiversity. Because if you try to eliminate one of them, you suddenly have eliminated a whole chain of interactions between these things. Fusarium doesn't exist by itself. If you take it out, you have broken down a very complicated, intricate system of interconnections. And that's, I believe, the most important lesson one might take. Always go for biodiversity. So, how do we encourage these fungi in soil, and specifically endomycorrhiza, which are so great and they do so much for us? Well, you can go and buy them in the store. They have them. They sell quite a few species, around 12 I've seen in the market. Either you buy 1 or 2 or 3 or a bunch of 12, which is a lot more expensive. It's 100 to 160 per kilo, which is enough for 0.1 to 1 hectare, depending on your use. Or you can just reproduce them yourself. Apart from the fact that they are much cheaper, free, it just costs you a couple of hours of work, not even, they are much, much more effective. Why is that? Even if it can be the same species, but there are different strains. Humans are the same species, but some of us are more adapted to living in Canada, some more adapted to living in the desert. Some are more adapted to living in high altitudes, some are more adapted to living in the sea, or spend more time in the sea. We're the same species, but we're not all the same. We have different abilities. So even on the same species, some of the mycorrhiza that have been developing in this environment for millions of years, they are much better at doing the right job in these conditions. When you produce a strain in North America and you're importing it here and you put it on your tomato that you plant in Ceyana, maybe it doesn't do anything, maybe it doesn't survive, maybe it will just do 1 or 2 of the things that you need. So it's much better to have native mycorrhiza. Also, the diversity. As I told you, you can have up to 12 I've seen commercially produced. If you take a scoop of healthy soil from the forest, you might have 100 different species there. If you're able to reproduce these, it's much, much, much more efficient. And there are many, many studies. This is one of them. They tried allocating and commercial actually mycorrhiza, single ones, multiple ones, and comparing them with native mycorrhiza in many different spots around the world. And they found that every single time the native mycorrhiza that they reproduce with the way that we're going to do it today was a lot more efficient than any commercial product. So it's something free that's much more efficient than commercial. If you're bored or lazy or you don't have the time, maybe you can go buy some. But it's so simple, as you'll see this afternoon, that really there is no need to ever buy any mycorrhiza. It's extremely simple and very efficient way. The last thing, very last thing, because I read this and I found out about it just a few days before coming here. There is one study and one replication in the field where they realized that it's very recent, we thought that endophytic endomycorrhiza, they needed to actually, even to sporulate, you have the spores that they have created inside the soil, and to sporulate, what actually happens in nature is that the plant is exuding some signature molecules that are making the spores of the endomycorrhiza germinate. So it's the other way around. Before we said the ectomycorrhiza, they germinate by themselves, sexually mate, and they start looking for a root in space. And sometimes they find, sometimes they don't find. They keep on eating until they find something. The endomycorrhiza, the mechanism that we knew is that the plant is exuding some substances, and these substances are triggering the spore to germinate. And so it knows that somewhere close there is this guy. But recently they did this experiment and they found that with isopropyl myristrate, which is a very common substance, it's you can find it in nutmeg butter, in coconut oil, in palm oil, many of them. It is actually one of these trigger molecules. You can put it in your compost, and then the endomycorrhiza will sprout and live even if they have no partner yet. It's very expensive to buy, it's 90 euros per kilogram, but it is a pretty exciting kind of development because we're starting to discover how these endomycorrhiza meet their hosts. And this way, maybe we can go one step farther and actually trigger beforehand the mycorrhiza to grow before we actually plant our actual seedlings. So that's the latest development. And last, you can exit the presentation. Yeah, that was the last. So we'll go do this afternoon some compost to encourage our fungal soils, and afterwards reproduce a mycorrhiza so that the people here, you see the process, and the people here will have a lot of inoculum for their plants in the next few months. That's it. If you have any questions or contributions, we have some time, I think.
Yes.
I heard that the endo, the spore, because it needs the plant, it has a limited time since it's
Absolutely.
to get to a root. So
yes.
Maybe that is good to do it just before you...
Exactly, that's the thing. Yeah. Right before you plant it, you maximize probably the associations this way. Yes?
Can you say something quickly about like fungal culture and mycorrhiza?
Fungal culture and mycorrhiza. I can say something quickly.
Something that develops mycorrhiza, like this process?
Well,
it does.
look, fungal culture,
Yeah.
I mean, most of the things we said in the first part about how to encourage fungal soil is in hugelkultur. You dig inside and you put a lot of lignin and a lot of woody material. And then this way it keeps wet, it increases the porosity, it has food, so it encourages. Specifically about mycorrhiza, I don't know, but I know that it encourages fungal life a lot.
Yeah, this I saw in my land, but yeah.
Exactly. Specifically about mycorrhiza, again, they do exist here and there, but in degraded lands, they don't most of the time. And we saw the reason why, where they're coming from, right? And if they didn't have host plants, they're not going to be there. The spores do survive for a few years, but if for 30 years it was degraded land, they're not going to be there. You have to bring them in.
Okay.
Yes?
So the idea is that we can use the native mycorrhiza for the plants we are going to put to the soil, to plant, to enhance, to enhance them. For the plants that already exist somewhere, what do we do?
Yeah, good question. Well, first you can also, if you plant close to them same or similar species and that are already inoculated, the mycorrhiza will grow and they will sporulate, and then next year or after a couple of years, they will find their way. I think the best thing you can do is this. Some other people I've seen that they are actually digging, disturbing the root system and throwing spores in there. I think it takes more energy, and what you have achieved is less than if you would actually plant an already inoculated plant, but both strategies can work.
Okay, and in the same sense, how could one know that from the community of plants we have, let's say here, that is a regenerated ecosystem, that indeed the mycorrhiza there are the right ones, or that that it's good to take from this plant and not the other plant.
So,
Stuff like that. Any criteria?
I don't know if you were when we were collecting some samples. So, we go with families. They're not very strict again, these things, like there are mycorrhiza that can associate with different families of plants. But again, going into the strain understanding, it might be the same species, but not the same strain. So, in general, you can find the mycorrhiza that associates, let's say, with grasses and with allium, garlics, onions, etc. But this specific strain that has been developed over thousands of years or millions over here, maybe didn't have any alliums here and has better association with grasses. So you go around, and if you want to cultivate grasses like corn or wheat or whatever, you go around picking grasses, wild grasses. It's much more probable that these kind of strains will help both of them, wheat and the wild grass. If you want to cultivate garlic, you go around picking wild allium and taking the spores from there. It's much more probable. The key there, as long as we're not in a lab where we can isolate this and that and this, and again, in the lab, you might recognize the species, but you don't know unless you do a DNA analysis that is this strain that is specifically for this kind of plants in this environment. The key is again, the answer is again the same, which is biodiversity. Diversify. The more you get from different sites, from different species, the better.
It's very likely it's better to go for the wild variety.
Absolutely.
What you want to plant rather than the domesticated.
Absolutely, absolutely. Because even in very healthy gardens that look very healthy, it doesn't mean you have the mycorrhiza there. It's more probable than in a dead one, but if you have brought compost from somewhere else, let's say, it doesn't mean that you have the right mycorrhiza. So it's always helpful. Encourage, put more and more and more of species. And again, I advise from close to your area, because again, the conditions are more right. Jorge and then Yorgos.
Yes, I understood from the presentation that some ecto, they produce some fruiting fruiting bodies.
That's right.
And some of them may be interesting for human consumption. What will be different than, for example, what we saw yesterday in order to get those fruiting bodies now that the case is mycorrhizing and not like the
For cultivating them, you mean? Well, you would need a host plant in order to get the fruiting bodies. So they're doing it now. They've started doing it with, I think, with oaks and other examples. With the Quercus family. I know that they plant the tree already inoculated with the specific mushroom they want to eat, and they plant them, and after a few years, they start producing. Yeah. It's more work, it's more patience. You need to wait for that to happen, but then you can have a supply for many, many, many years, because it's not like a substrate that is being eaten, it's consumed, and it's over. It is a constant kind of association that you can be eating for many years after. It's an investment in time and energy. Yorgos.
Yes, so a question in the same group, I think, that of the questions that Theodoris asked is like, so I show up in a field and there is soil. How do I estimate what's the fungal reserve there, fungal capacity that's already there? It's like, take some grass out, and what do I do?
So, the easiest way to guess is to see the health. If it has a lot of organic matter, if it has very simple way to do that, not accurate, but in the good direction, how dark your soil is. Because there are some dark soils that don't have organic matter because they're dark because their mother, the bedrock is like that. But general rule, the darker your soil is, the more organic matter it has. The more diversity it has. If you count the species over there and you find 2 or 3, I'm pretty sure it won't have a lot of fungal life. If you find in 1 square meter, 20, 30, 40 species, I'm pretty sure there is a lot of fungal life. If you find more later in succession kind of plants, like big trees and bushes and stuff like that, there is fungal life, almost certainly. If you find orchids, it's 100% sure you have fungal life. So depending on, again, the whole image. You can go in the lab, of course, again, there are very simple tests where you can take one little sample of soil and actually count in a specific volume how many fungi, how many protozoa, how many bacteria you have. And this is a good indicator. This is one of the processes we do in order to actually consult, if we have a microscope, and to consult a farmer what they're missing. It's easy, but you need a microscope. Otherwise, by looking at the whole image of what's growing, how is your soil, you can guess pretty well what's going on underneath.
The grass that we took out yesterday, the different plants, could we see the, mycelium?
Endomycorrhiza, you cannot see. Ecto, tomorrow we're going to see a lot. Like just lifting a little bit of organic matter, we'll see it growing everywhere. Tomorrow we're going to do the walk in the forest. You had a question?
I think it was Maria, maybe. I don't know. I was just was going to ask, like, if we want to plant like trees, what mycorrhiza will you use?
For endo, we're talking about now?
Yes.
Again, I would go to the forest, dig, take, because again, you have to dig a little bit down, especially endomycorrhiza. You have to go 30 centimeters and under to increase the count of spores. So dig, take some soil. If it's close to the root, try not to disturb it as much as possible, but take soil from around the root of similar species. Like if you want to plant a fir, then go to a conifer forest. If you want to plant an oak, go to oak forest. And get samples from there and do the process that we'll be doing this afternoon in order to multiply them. Maria? No, you have nothing. Same question. Say it. Okay. Anything else? Ready to get?
Is there anything that we can use in compost or like directly on the soil to increase the activity of mycorrhiza in a garden that is already established or something like that?
But you say in compost, and there is already so compost to make compost to put it in an established. Well, the fungal compost we're going to be making is probably the best thing you can do, and we'll talk about how we can be using that compost because there are different ways that we can multiply it actually. But the fungal compost we'll be doing later on is the single best thing to do for encouraging fungal life.
Any cheap tricks? For example, like if you want to help an entire mountainside or like a...
Yes, we'll say in the afternoon.
Okay.
Basically, you make this compost, I'll tell you now. You make this compost, you've made compost tea before. You do something very similar, but not with molasses only, because molasses are encouraging mostly bacteria. With other stuff like fish emulsion, etc. You say make a compost tea from that kind of compost, and you're multiplying for a few hours. One thing, and maybe it's an opportunity to say now, many people are pretending to do compost tea with a tiny little aerator that they use in aquaria. This doesn't really work. That's not enough oxygen there. It goes anaerobic really quickly. You need a lot. Like if you see a real compost tea that is made, it's done, like you think there is a volcano coming underneath. It has to be very well aerated in order to encourage aerobial life and multiply it really quickly. So that's a way to do it, and then you go spray. With a few kilos of compost, you can really cover a hectare. You can multiply it a lot, and it's very effective. That all?
Can I ask one?
Last question.
Then I have to make an announcement.
Okay.
Sorry. About the plants that you use, you talked about to absorb the lead and like, can you compost them after?
Absorb? Sorry?
Sorry, the plants that you use to absorb.
Oh yes, heavy metal.
Compost them after to neutralize the.
No, because the lead will be there.
You have to. Yeah.
You have to remove it,
Okay.
bring it to a specialized kind of laboratory where they will break down the plant, burn it probably, and remove the lead to reuse it, because they're very useful heavy metals. They're bad when they are in great concentrations, but they're very useful for a lot of applications.
But then you need to find a lab that can process these waste.
Yes, in Greece I mean,
Yeah.
I don't know of any, but
Mine does it.
it does it.
Yeah, they do it in lab now.
Ah, amazing. That's amazing.
This is because they could not use like the soil that is already there for the experiment. They firstly go and record all the species of mushroom you find there. Speaking of ectomycorrhiza, and then these species will be cultivated in some blocks on the soil, and then this will be also attached to the soil, and then all this is removed and it's burned in a way that all this
They extract the heavy metals.
exactly.
Really
Wow,
nice.
that's amazing. I
Cool.
never